Okay, so just to repeat the methods till the rest of them join. So there are three methods, three broad methods of transcription. One is the chemical-based method of transcription. The one is physical-based and biological method of transcription. In chemical-based, you're going to use different chemicals which will start interacting strongly with the DNA molecule, with the negative charge of the DNA molecule, and help it to enter the cells, target cells. Instrument-based, you're going to use physical instruments such as a little bit of current, yes, physical properties of certain instruments. Even the current is delivered to create force on the surface of the cell, and it will allow the DNA to enter the cells. Okay, and ballistics method, you're going to use certain tiny metal particles onto which your DNA is going to be coated, and a gun is there to create a momentum movement. Yes, a momentum force which will force the bead which is coated with your DNA to forcefully enter the target tissue. This ballistics method is usually suitable for delivery into plant tissues or animal tissues. Even animals are actually fired. They are placed in a small chamber, and it is subjected to continuous firing with what to say, the gene gun with different DNA molecules. It is quite very sad to see that. But one of my professors was saying that even that is done, firing the animal with your particle coated with your DNA. And laser fection or opto injection, where it is the laser is used, okay, for the method. And the last method is virus. So you allow a virus, okay, to infect your target cells or target organism, and this viral particle which is going to infect your target organism, target cell, okay, that will not be able to deliver your gene of interest unless and until it is assisted by a helper phage. The helper phage will help this viral particle, this recombinant viral particle, to successfully infect your target cell. Now let us look at all these methods. Today's class will finish up the chemical methods. So in the chemical methods, yes, so chemical method of transfection, it is a popular technique due to ease. The cost is very, very less, and wide variety of transfection reagents available. In this method, what happens is that your DNA molecule, it will start interacting with your reagent. So liposuction also comes under the technique of the chemical methods. In liposuction, what will happen? You're going to have different cationic lipids. What are cationic lipids? They're nothing but phospholipids. So can anybody name which are the phospholipids? Can anybody name which are the phospholipids you have come across till now? Biomolecules have studied about phospholipids, right? Yes. Vaishali, can you name any phospholipid? What is there in egg? Phosphatidylcholine, correct? So phosphatidylcholine, what is the, structure of choline? Let me write the structure of choline. So it is CH2, CH2. Sorry, I'm writing on that thing only. N, CH3, CH3. Okay. The valency of actual nitrogen is actually, though it has 5 valence electrons, by actually having 3 covalent bonds, it becomes stable. The presence of an extra covalent bond, yes, will render it a positive charge because the electron is shared with this third methyl group which is being present over here. So that is the reason why this nitrogen atom, which is part of the choline functional group, it becomes positively charged. And because of the presence of this positive charge, phosphatidylcholine is positively charged. So therefore, this kind of the cationic lipids, what I was mentioning, what will happen? They will start interacting with your DNA molecule very well. Okay, you understand? So the negative charge over here, the negative charge of the DNA molecule will start interacting with the positive charge of your lipid molecule. And because of the stable interaction, you can direct this complex of your DNA along with your chemical to enter the target cell. Okay, so calcium chloride is used. Why calcium chloride? Because the calcium cation, it will start interacting, it will start stabilizing the negative charges of your DNA molecule. Polyethylene glycol can form hydrogen bonds with your DNA molecule, not dealing with the nitrogenous bases, obviously they will not. they will start forming hydrogen bonds with your hydrogen atoms of your deoxyribose sugar, or even with your phosphate groups of your sugar phosphate backbone. And DEAE dextran, nothing but diethylaminoethyl, I already told you. The amino group, because of the presence of the positive charge, that will start interacting with your DNA molecule, and that is coupled, this diethylaminoethyl, it is coupled to a polymer. It's a natural polymer. The polymer will make sure that it will start interacting with the cell membrane phospholipids and stably enter into the cell. Yes. And the last one is this liposome mediated transfer. That's what I mentioned. Yes. We'll study about all these methods in this class. Now, calcium will start interacting with the sugar phosphate backbone. Yes. And along with that, you also have your diethylaminoethyl dextran, two different strategies. So that will start interacting with the negative charges. Each of this different method as used over here, this is the method of liposuction. So in all these three different methods, what you have is that the reagents will start interacting with your negative charges of your sugar phosphate backbone of your target DNA. Yes, it will stabilize that. Once it stabilizes that, it will be able to safely deliver to your DNA molecule into the target cell. Okay, so let us say you have to meet somebody, right? You have to meet some very important person who can do a favor or something like that. Somebody should introduce you to this very important person. Otherwise, you will not be able to meet him. Let us say you have to meet a big shot. Let us say you have to meet Mukesh Ambani. Yes, somebody has to meet Mukesh Ambani. Let us say I have to meet Mukesh Ambani to develop a business. Do you think Mukesh Ambani will give me an appointment without any this thing contact? No? Okay, I have some idea, but Mukesh Ambani will not believe me. Okay, and let us say Priyanka and Vaishali. Let us say for some reason you have done internship at Reliance Industries. Okay, and for some reason Mukesh Ambani has personally seen your project work, and he's impressed really by that. And you, both of you can come and have a discussion anytime. So I'm your, let us say, professor, I would like to interact. He will not give me an appointment. If you go and tell somebody, as one of my professor, what to say, faculties wants to speak, then you will be able to, I will be able to meet him. Understood? Okay. It is something like that. If you have to meet some person, you have a contact which will help you to meet that person. These molecules are very much similar to that. Without that, you will not be able to deliver your target DNA. Okay, so that is what I remembered right now. How to explain these chemical reagents. Chemical reagents, they will interact stably with your nucleic acid and help it to deliver into the target cell. It could be either calcium phosphate, it could be D8-dextran, or polyethylene glycol, or your liposomes. Let us look at each of these methods. In calcium phosphate or calcium chloride method, what will happen? First, you need to mix calcium chloride with DNA in phosphate buffer. That will lead to the formation of calcium DNA calcium phosphate co-precipitate. Now, what will happen in your sugar phosphate backbone? You're going to have the negative charges, P O minus O minus. That will start being stabilized by the divalent calcium ions, and of course, phosphate will also surround the divalent calcium ions. This results in DNA and calcium and phosphate forming a precipitate, co-precipitate. So these precipitates are mixed with your target cell. Okay. So when it is mixed with the target cell, through phagocytosis, this entire thing is taken into the cell. Okay. So once it is taken into the cell, later what will happen? Your target gene will integrate into your, it's quite possible if it is a stable transfection, it will get integrated into your genomic DNA. Now, this is more efficient as calcium will give more uniform particle size. Polyplexes or polycationic complexes that form soluble complexes, also called polyplexes, through spontaneous interaction with the DNA. So most of these cationic polymers which are there, yes, they will start interacting with your DNA. Once they start interacting with your DNA, that is when it forms a polymer complex. Cationic polymer complex with DNA, it is called as polyplexes. Okay, so that will help your target DNA to enter into the cell. So this is what is happening over here. You have a complex polymer interacts with your DNA molecule. It will form a particle Also, can it as a nanoparticle, if you know its structure properly, if the structure is stable every time it is being formed, that will help in its delivery into your target cell. Once it enters the target cell, usually this enters only this method. Only please don't, it is only this method is through phagocytosis. This was the question asked to me in my seminar, not with respect to calcium phosphate. With respect to electroporation, yes, people asked a question, how is the DNA delivered into the cell? That's the professor, he tried to help me. He said, are there any holes being formed, any pores being formed in electroporation? I said, no, sir, it is through phagocytosis. It's very silly, the thing what I answered. Phagocytosis is only in the case of when it forms a complex with calcium phosphate. Because the particles, the cells might recognize them as something as some nutritional source, and they'll try to engulf them and put it within the cells. Once it is within the endosome, under the influence of the fusion of the endosome with the lysosome, it will form the phagosome with the lysosome, it will form the phagolysosome. So what will happen? The DNA gets released into the cytoplasm. And it quite happens that this dissociated DNA will slowly enter the nucleus and get integrated. It's one of the mechanisms by which this is done. Okay, but there is always a chance, students, please remember, during this phagocytosis, your target DNA might also be damaged during this process, because the hydrolytic enzymes which are present, they don't leave spare any DNA molecule. They are known to degrade proteins, but they cannot be very lenient towards DNA. It quite happens that they could degrade your DNA also. So you need to make sure that this doesn't happen. Okay. So this was a question asked to me, which I did not answer it properly. One of the questions, two questions were asked, I didn't answer them properly. One of the questions was this, that in the case of electroporation, what will happen? I said, so they'll enter through phagocytosis. It's not actually correct. This is actually through formation of pores on the surface of the cell. Okay. Now, apart from this, we can also use what is a cations based, so reagent based, where we will start interacting. Okay. So lipid mediated. So in lipid mediated, so which is this? Okay. A lipid mediated, as you can see, the structure of the lipid molecule over here. The structure of the lipid molecule is in such a way that you have CH2O, one fatty acid, first carbon atom, and CH2O, C double bond O, and the other one is, I'm sorry, that is CHO. Yes, this is what is this? It is present at the center. Is it? Okay. This is something different. Dialyl phosphatidyl ethanolamine. So the presence of the the phosphate group is at now second carbon atom instead of the third carbon atom. This is to make sure that the lipid molecule has a specific symmetry. So you know that most of the lipid molecules, CH2O fatty acid, CHO fatty acid, and CH2O phosphate, and the nitrogenous base, or whatever specific functional group will be there, choline or ethanolamine or whatever it is. In this case, it is quite different. These cationic lipids used for the formation of liposomes. So in this case, what happens is that the second carbon atom is having an ester bond with the phosphoester bond, and this has another ester bond, yes, the phosphate group with ethanolamine. Okay, so therefore it is diolyl phosphatidyl ethanolamine. So why diolyl? Because there are two oleic acids which have been forming an ester bond with the OH group, yes, CH2OH groups, yes. And the second carbon atom, it is having a phosphoethanolamine. So what is ethanolamine? So what is ethanol? CH3, CH2OH. So this is CH3, CH2OH. Ethanol amine, yes, it is nothing but phosphatidyl ethanolamine. In this case, it is diolyl phosphatidyl ethanolamine. In this case, the phosphate atom is actually, the phosphoethanolamine group is not on the third carbon atom. Instead, it is there on the second carbon atom. So therefore, what will happen? This will form the polar head, and this will form the hydrophobic tail. So this is also called as dope. Okay. So this is used to create liposomes. Once the liposomes are created, yes, they will start to interact with your DNA molecule. So what will happen during the interaction? Your heads, this positively charged groups, this will start interacting with the sugar phosphate backbone and help in the successful complexing of the DNA molecule with your liposome, that is your lipid vesicle, and this lipid vesicle will be able to successfully integrate with your target cell. How? Because these are cationic lipids, they will have affinity towards the phospholipids of your cell membrane and help in the transfer of your target DNA into your target cell. Yes, target eukaryotic cell. Now, how is this done? Yes, this is, yes. So, I think this is a different method. So, lipoplexis. Okay. Yeah, so this is what is happening over here. Your target DNA, yes, your target DNA is added to the liposome, which forms a complex lipoplex. So we had studied about polyplexes, right? That is in the case of polymer. This is not a polymer, instead it is a lipid. So this will form a lipoplex. The lipoplex will fuse with the plasma membrane, yes, and lead to the formation of this lipoplex endosome. This endosome will have the lipoplex within that is later released, and the DNA gets released into the cytoplasm and later goes to integrate with your genomic DNA. The advantages is that deliver nucleic acids with greater efficiency. Yes, easy to use, minimal steps required. Works in a wide variety of eukaryotic cells, can be used for in vivo transfer also, not only DNA, even RNA also, and successful delivery of DNA of all sizes, okay, from ranging from different polygonucleotides to several kb, okay, and even in the yeast, artificial chromosome can also be delivered using this method of lipid, liposomes. Disadvantages: this is not applicable to all cells. Why not all cells? Because only animal cells can be used. So plant cells, you need to create protoplast and after that use liposuction. Otherwise, it's not very stable. Efficiency will be sometimes low in primary cell lines. Okay, is one of the disadvantages. Now, this calcium phosphate method that I told you, the first DNA is mixed with calcium chloride. Yes, it is incubated, the target cells with it, and as I told you, so that calcium phosphate is engulfed through phagocytosis. So this method, whatever indicated over here, this slide is for the transformation, bacterial transformation. So bacterial transformation, what is going to happen? You're going to resuspend your bacterial cells in calcium chloride. Yes, and you're going to add your DNA, and the DNA will form a complex as a polyplex. In this case, it is not a polyplex. Calcium chloride will start interacting with your DNA. Yes, and because the cells are competent, pores are created on the competent cells. So once you give a heat shock, the pores will expand in size, and your plasmid is able to successfully enter your bacterial cells. Then you cool it on ice, the pores will seal once again. Once they open, the DNA enters, it will close. Keep it on ice for some more time, and then you add LB to stimulate the division of your bacterial cells so that they also start producing the antibiotic, and then you rate them. This already you have done it in the lab, that's very good. Okay, so you know this procedure. I'm not very particular about telling it once again, so I'm not doing that again. So pros and cons is that advantages: calcium phosphate, really available. So it can be used for both stable as well as transient transcription. If you don't want your gene expression to take place beyond a certain amount of time, so for the transient transfection, you can use that. The calcium phosphate appears to provide protection against all intracellular and serum nucleases. Serum will also have nucleases. If you are using serum-based medium, okay, for the animal cells, you can use calcium phosphate. But sometimes what the disadvantage is that the efficiency is low. It is not suited for in vivo transfer. It is only for in vitro transfer, only into certain cell types. Yes, only into in vitro. So it cannot be used for in vivo. For in vivo, the only method known is this method of microinjection. Okay, even virus also can be used, but microinjection is preferred mode. So sometimes what happens, calcium phosphate may be toxic to cells. The primary cells will not be able to grow. And if there are certain changes in the pH and the ionic strength of your buffer being used, this might not work very efficiently. Cationic polymers. In the cationic polymers, you have a hydrophobic group, yes, and a cationic group. There are two different parts of the same molecule. That as polybrine, yes, you have the polyethylene imine and dendrimers. So they're able to form a polyplex along with the DNA molecule and help it to enter. As you can see over here, what is happening over here? Yes, your cationic polymer. Can you see this cationic polymer? Why is it a cationic polymer? Because it has several positive groups on this chain. Yes, and this is supposed to be the polymer. So this cationic groups, they will start interacting with the sugar phosphate backbone, and this polymer will help in the integration across the plasma membrane. You know that anything which has to cross the plasma membrane, it has to be hydrophobic. If it is hydrophobic, it will interact better with the plasma membrane. It will help in the integration within the cell. Okay, that is about the cationic polymers. Okay, how it is coated onto the surface of the cationic polymer, and here it is the cells. Dendrimers are highly branched globular macromolecules capable of interacting with DNA to form small complexes. Positively charged amino terminus on the surface of the dendrimer. The dendrimer is usually a synthetic molecule which is created, which has different amino groups on the surface. They will start interacting with your, your DNA molecule, and this entire complex is used to help the DNA enter the cell. Now, D8-dextran, diethyl aminoethyl dextran, as you can show you here. Now, this is ethyl, diethyl aminoethyl. So, this entire group is diethyl aminoethyl. Okay. So, dextran just formed a bond. Just formed a this thing. In this case, it is a kind of bond. Glycosidic bond which is being formed by the ethyl group, ethanol group, and dextran. Okay, so this molecule will help to interact with your DNA molecule. This positive charge will start interacting. This is inexpensive, easy to perform. Yes, and be applied to a wide range of cell types. And this can be used only for transient transfection only, not for stable transfection. Sometimes the efficiency will vary. Okay, so as you can see here, please students, please take a note of all these diagrams. It might be helpful for you, and how DNA gets complex with these molecules and results. Please, this is quite important for the examination point of view. This magnet-mediated transfection. Magnet-mediated transfection uses magnetic force to deliver your target DNA into the eukaryotic cells. So here, nucleic acids are first coated with magnetic nanoparticles. DNA are bound to the magnetic nanoparticles, and which move from the media to the cell surface by applying the magnet force. So as you can see over here, your DNA molecule is covalently bound to certain magnet nanoparticles, and it is placed in close vicinity of your eukaryotic animal cell. And if a magnet is used, it will pull. Let us say if this is the eukaryotic cell, you have this could be, let us say, your target DNA, which is covalently linked to your magnet. And if there's nanoparticle, magnetic nanoparticle, and if you use a strong electromagnet, it will pull this nanoparticle into the cell, and that is when your gene of interest will be stably transferred to your target cell. Okay. In this case, it is very rapid. The efficiency is very good in the case of transfection. High efficiency rates for adherent cells, and primary cells can also be used for this, and it can be performed in the absence of serum also. So the main principle what is involved over here is only the use of a magnetic force, which will force your target DNA to enter a certain cell. So disadvantages, it requires adherent cells. Suspension cells is difficult to get transfected by using this method. Okay, magnet transfection. So biophysical based method. So this is nothing but your you have the biolistics method. I'll show you the slides later for this. Now, electroporation. This was first reported in mouse cell lines. This is a technique wherein you have protoplasts that are created from plant cells or the animal cells. Okay, a small electrical impulse is used in order to create certain pores on the surface of the cell. Okay, because of that application, this you need to standardize in the lab. How much charge should I apply to make sure that there are pores which are formed on the surface of the cell? When you do that, you find that your DNA molecule will enter your target cell. Okay, after that, this electricity is switched off, and then you can see whether your DNA has directly entered your target cell or not. So it is a very fast technique, electroporation. Please remember, it is not given for 2-3 seconds. No, microseconds. You will not even be able to visualize how fast it is happening. Just need to, you will have electroporation cuvettes. You need to add your mixture of your DNA along with your cells. Keep it in the electroporation cuvette and keep it within the chamber. it will give a pulse which is mediated by the software, let us say for 30 microseconds. With 30 microseconds, or let us say 300 milliseconds, your DNA will enter the cell. You won't be able to see that, oh, is it over? It's not that easy. That is the way. But only this other disadvantage is that you require instrument setup, you require some finance to set up this facility. Once it is set up, it is quite easy. I too have done this in one Professor Parag Satalesa. He passed away, he was in IAC. So he allowed me to use this facility in the year 2009. So we had tried to transfect, what to say, a particular plasmid construct into, what to say, plant cell line. It might sound very silly. It is called as the bright yellow 2 cell line, which is actually a plant cell line. It requires totally plant growth medium. Growth medium has to be completely plant based, but it is called bright yellow 2 cell line because people started comparing with tumor cell lines. Okay, so the disadvantages of this method is that cell mortality will be more, efficiency in primary cells, and high mortality rates caused by the high voltages. Because electric pulse is applied, it quite happens that the electric pulse might kill most of the cells. Okay, so only unless and until you know that your primary cells are not killed, okay, you will be able to use electroporation. Okay, electroporation should be used only if it doesn't kill the cells. Current has to be standardized, whatever current you're going to use. Standardize the current and make sure that it doesn't kill the cells. You can use it for this. That is the reason for electroporation. First, you need to standardize how much of pulse is applied, for how long it is going to be applied. Once you standardize only, you'll be able to do that. Now, this is another technique called cell squeezing. It was developed in 2012. So what you do is, you take a target cell and start squeezing it, okay, through a given small capillary region. Okay, when you squeeze this, what will happen is that there will be temporary disruption of your cell membrane. When squeezing occurs, what will happen? The phospholipids of the cell membrane will slowly get destabilized. At this point of time, if you introduce your DNA, it will try to enter the cell. So this is called as transfection by cell squeezing. This was discovered by Armand Shari, Robert Langer, and Klaus Jensen in the year 2001, recently, about 8 years back. So this eliminates the possibility of toxicity. So this will not kill the cells. So, transient holes are created because of the distortion movements. It will help to transfer your DNA. Okay. So this is about the squeezing procedure. As the cell reaches the squeezing area, so what will happen? It will be exposed to the DNA molecules, and this exposed DNA molecules will be able to enter the cell. So this is the biolistics method. I'll teach you about that later. Okay, there's one more PowerPoint which is also nicely given. First, you take your gold particles and coat it onto DNA. After it is coated onto DNA, the particles which are coated with your DNA are loaded into cartridges. The cartridges loaded finally into the gun, and you fire your tissue. When you do this firing, your target DNA enters the cell. Okay, I think another 10 minutes, students, 10 minutes remaining, will end the meeting. So in this method, so you have an acceleration tube. Yes, you have the macro carrier. So this is the target gun. So as it keeps, what to say, entering the stopping screen over here, you can see that a magnetic field is also applied sometimes in order to accelerate this movement of the whole particle of metal ion, and used to bombard onto your target tissue. The simple and rapid technique, it is cell type independent. It doesn't depend on the cell onto which you have to do this transfection. It can, one second. One second, students, just 1 second. Students will stop the class here. I think we have a meeting now. So I'll stop the class with the biologistics delivery. Okay, so the advantages of this method, it is a simple and a rapid technique. It is cell type independent, doesn't depend on the tissue. It uses small amount of DNA. It can deliver large, what to say, quantity of DNA. Each particle is coated with small amount of DNA, but since you're using many gold particles, you can deliver large amount of DNA. So the chances of success are quite high, and for this tissues are used for this, and reproducibility is quite high. Okay, disadvantages is that the cost of the gold particle will deter you from using this technique. Okay, and preparation of the particles involves a lot of skill also, a lot of cost. Okay, and it generally there is lowers the efficiencies compared to electroporation. Sometimes it depends. Okay, so this is difficult to use this technique for single cells. For tissues, yes, it is a good method. So instrument cost as well as the gold cost, that will prevent you from using this technique. So sometimes it causes physical damage to the organism, this method. So students, I think